This month's focus feature in the Journal of Hepatology addresses two very interesting studies looking at both the mechanisms and detection of fibrosis. In the first study by Choteau and colleagues from France, they examined a transgenic mouse model previously developed by Dr. Stanley Lemon, which overexpresses the hepatitis C viral genome in hepatocytes of transgenic mice. The new angle on this study was that in addition they induced mild liver injury due to carbon tetrachloride, which is a standard hepatotoxin. Interestingly, they found both that CCL4 in combination with hepatitis C viral proteins greatly accelerated the development of fibrosis. It also induced progenitor cell expansion, which are two very interesting findings. First, because they suggest that a mechanism by which hepatitis C may synergize with other forms of injury, which in humans could be alcohol or obesity. And secondly, because it adds further evidence to the idea that as livers are injured, progenitor cells respond and perhaps lead to regeneration. There is still a mystery between the pathogenesis of injury and how it links to progenitor cell activation. Nonetheless, this provides fertile questions for further evaluation. The second study by Polasek introduces a potentially important new diagnostic approach to quantifying fibrosis non-invasively that may ultimately be applicable in humans. Specifically, they've identified a chemical gadolinium-based contrast agent that can be used to quantify collagen in an animal MRI methodology. So they inject this contrast agent, and the amount of binding to the liver is proportionate to the amount of fibrosis. Now, this was a preliminary study, and much further evaluation is required to ensure that the amount of gadolinium binding and therefore the image generated by MRI are directly proportionate to the amount of collagen. But in the best of all possible worlds, this could represent a new MRI-based technology that could quantify collagen precisely in human livers, thereby obviating the need for liver biopsy. Still, as I noted, there are additional studies that need to be done in animals, much more extensive quantification of binding and its correlation to fibrosis. And it also raises the question of whether just measuring the amount of fibrosis alone in the liver tissue will be sufficient. After all, we know that changes in fibrogenic activity occur much more quickly after improvement in liver injury than the absolute amount of scar. And so if we really want to detect changes in fibrogenesis, we're going to need methods that complement this potential approach of quantifying fibrosis. And those new methods might include assessment of stellate cell activation or other measures of fibrogenic activity that we would expect will change before fibrosis content itself is altered. Nonetheless, these two studies are really important in telling us that we are moving closer to the idea of both understanding the pathogenesis of fibrosis and, more importantly, detecting it using a non-invasive method. And we think that these are two among many very important studies in advancing the field of hepatic fibrosis and translating it into new therapies.